also been said that the TB places its highest burden on the world's most poor and vulnerable, aggravating existing equivalent to more than 50% of their income on average. According to the same WHO organization, or WHO we can call simple, 2022 global report, an estimate of 106 million people fell ill with tuberculosis worldwide in 2021. Out of these, about 6 million men and 3.5 million women, and as well as 1.2 million children. TB is present, thus you can see from the previous figure that TB is present in all the countries and age groups, but it is curable and preventable, which is the best part. In 2021, the 30 high TB burdened countries accounted for 87% of the new TB cases. So if you look at in here, we have 44% in the Southeast Asia, 25% Africa, 18% Western Pacific, and then 7.7% Eastern Mediterranean, and then we have 2.8% in America and 2.7% in Europe. So this is the area where we have maximum incidence of the TB. Besides that, we have also the problem of multi-drug resistance, MDR-TB. It remains public health crisis and health security threat for some time. Only about 1 of 3 people with drug-resistant TB accessed treatment in 2020. So situation is not very good. But at the same time, efforts are being made to take care of TB as much as we can. So globally, TB incidence is falling at about 2% per year, and between 2015 and 2020, the cumulative reduction was about 11%. This was over halfway of the end TB strategies, which has been set by WHO and then United Nations. It is a TB strategy milestone of 20% reduction between 1950 to 2020, and we have only 11%. An estimated 66 million lives were saved because of the emergency, what we can call measures, saved through TB diagnosis and treatment between 2000 and 2020. Ending the TB epidemic by 2030 is among the health targets of United Nations, as mentioned earlier, United Nations Sustainable Development Goals. So here we will discuss first the table 1, and then we will go to the table 2, that is related to the TB status in Pakistan. So what is the vision of this NTB strategy at a glance? It is vision is that a world free of TB, zero deaths, disease and suffering due to TB. Goal we have to end the global TB epidemic. Indicators are here, that is we have in this column we have the milestone in 2020, 2025, and then here we have the target 2030 and then 2035. So the first point is that percentage reduction in the absolute number of TB death. It has been compared with the 2015 baseline. So in 2020 it was 35%, 75%, and what target is 90 and 95%. Then percentage reduction in the TB incidence rate, again compared with the same baseline of 2015, 20, 50, 80 and 90%. Percentage of TB affected household facing catastrophic cost due to TB. It has not level 2050 was unknown, so that's why we have over all zeros. In 2014 and 2015, all member states of WHO and United Nations committed to ending the TB epidemic through their adoption of WHO anti-TB strategy and the UN Sustainable Development Goals. And the next, that is the estimate of TB burden Pakistan 2021, according to the same report of 2022. Total TB incidence in terms of numbers 611,000, and the rate per 100,000 population is 260. And then we have not only TB incidence, it is also related to HIV, MDR. So here it HIV positive TB incidence about 15,000 and 6.3. MDR and RR, that is the one of the leading drug for the cure of the TB, that is rifampicin. This is also people are getting the what we call the drug resistance because they are not following the advice what doctors gives for the treatment of the TB. So as a result of this, we have the incidence of 36,000 and then 15. HIV negative TB mortality is about 48,000 and then 21. And then HIV positive TB mortality is 2100 and 0.93. So this is the status of TB in Pakistan. Moving forward to the 2030 targets require the ensured availability of new tools from the research pipeline, in particular better diagnostics, including new point of care test, vaccines, safer, easier and shorter treatment regimens, and then safer and more effective treatment for latent TB infection, effective pre and post exposure vaccines. So we are working in this particular area, try to find out better diagnostic procedures or develop better diagnostic methods, test, and then able to develop specific vaccines which is not really available for TB patients. So next we go a little over the disease physiology. TB bacillus attack different parts of the body. Distinctive tubercles or tuberculosis nodules, small lesions consisting of dead grayish matter. Infection is usually from one of the three sources, that is infected milk, droplet infection from an infected person, or breathing infected dust. When it is when we think about the exposure of TB, then it could be possible that people get infected. The infection is only 10 to 30%, non-infection 70 to 90%. Out of this infection, 10 to 30%, it could be latent TB, 90 to 95%, or it could be active TB, that is 5 to 10%. And then within active TB, we have cavitary, and then we have non-cavitary. Besides, we have the TB. We can divide based on the organ, different organs. Pulmonary TB is 85% cases we have, and non-pulmonary, or what we can say extrapulmonary TB, pleuria, and then lymph nodes, bones and joints, skin, abdomen, genitourinary tract, and meninges. Individual who get infected with tuberculosis, and then this is pulmonary. A person may contract pulmonary tuberculosis from inhaling, as we mentioned earlier, droplets from a cough or sneeze by an infectious person. And as a result of that, there is a lesion one can see, that is what we say here is a granuloma in the lung tissue. Then we go to the genomics. The total genome of this bacteria is about 4.4 million bases, and then about 4,000 genes encoding protein, which has been determined by Cole in 1998. As a result of the sequencing of the MTB genome, we are able to figure out how many immunogenicity antigens are present. So there are more than 100 immunogenicity antigens are present there. So the scientists have thought that maybe they can use these antigens to develop different immunologically based diagnostic procedures. So single antigen has been used in different tests, but they are not much sensitive. That is what scientists have figured out. Rather than using single, it was thought that multi-antigen test should be used, which could differentiate active disease from latent and vaccinated individuals. Right now, we have the diagnostic methods available, that is AFB microscopy, tuberculin, skin test or Mantoux test, chest X-ray, DNA amplification by PCR, that is they are also called gene expert, bacterial culture, ELISA, microdot, but all these have some kind of limitation and shortcomings. So still the efforts are going on to see that how we can make more effective ELISA procedure. That is again antigen antibody based method to be developed, and multiplex microbead assay. This is a recently developed method for many other antigen antibase reactions. So we thought that maybe we can make the use of this one. So rationale was that is the recent advances have implication for improved diagnostics, since complete sequence of MTB genome and antigens are available. Multiplex assay technology enable rapid detection of multiple serum analytes such as antibody to multiplex bacterial proteins labeled with some kind of reporting molecules like a fluorescent molecule. So this fluorescent dye has also made, or in other words, it has provided tools to be used in different ways. So here it is. This is the basic procedure that is used when we try to detect TB antibodies in the serum. So here we have the antigen, different antigen. Here only 3 has mentioned, and then as a control, and then biotin also a little control, and then we have. So we take in 96 well plate. We have the whole instrument. In a minute we will discuss that, and then we have that the serum, patient's serum or maybe normal serum, whatever serum we are taking for in consideration. Then if any antibody is there based on the antigen, then it will bind. Then so this is the first antibody which is getting from the serum, and then we have to allow these antibody to interact with the second antibody, which is biotinated lab. And then how we are going to monitor this one. So we have a phycoerythrin labeled streptavidin, because here biotinated, so here is streptavidin, they have the affinity. So as a result of that, if anything is present over here, so we will get the signals. So this is the analysis mixture for bead identification and PE, that is phytoerythrin conjugated streptavidin. And as a result of that, you will be able to know how many different kinds of antigens are present as interact with the serum. And as a result of that, we will be able to find out some specific information. Here is the instrument which is being used for this method. The beads are red on a dual laser flow-based detection instrument such as the Luminex or BioRed Bioplex analyzer. As it's a flow-based, so it is basically based on the flow cytometry concept. We have two lasers. One laser classifies the beads and determines the analyte, and that is being detected. The second laser is the one that determines the magnitude of the PE drive signal, which is in direct proportion of the amount of the bound analyte. Here we have the comparison of multiplex system to conventional ELISA system for antibody detection. Parameter is given in this column, and the ELISA based on what parameter we are using. Here we have the specific remarks, and then here is for multiplex. So multiplexing limited for only 2-3 in case of the ELISA. Here we can go up to 100 analytes. So it is faster than what we have in ELISA. High throughput format for ELISA it is difficult. For multiplex, yes. Simple size you can have 50 to 100 microliter. Here we can go smaller, that is 30 to 50, and we can use for many analytes as we have already mentioned over here. Sensitivity 1 to 10 picogram per ml, the same over here. Dynamic range is 2 to 2.5 log, here to 2.5 to 3.5 log. Assay time 3 to 24 hours, here 30 to 90 minutes. Replicated measurements per sample 2 to 3, and here we have 10 times we can do. So that is so we can have better statistics to be taken care of. So here we have that. So with this background, now we go to the objectives of our research: to develop sensitive, specific, reliable, and cost-effective method for early detection and monitoring TB. It will enable the physician to initiate therapy sooner, to trace contacts of positive individuals more effectively, implement institutional procedures to prevent or reduce cross-infection. What was the strategy? So a strategy for serodiagnostics, blood samples collected from patients and healthy individuals per grouping. That is group 1 BCG negative, tuberculin skin test negative. Group 2 BCG negative, tuberculin skin test positive. Then group 3 active TB, group 4 reactivated TB. And experimental strategies was that we have selected these about 13 MTB antigenic proteins. And then we have done the optimization of expression of these antigens in specific, using specific vectors and then specific strains for the expression. Then we have confirmed the specific express antigen by Western blotting. And then we have done the bulk expression of each plasmid to be used in the multiplex as in ELISA assay. And then purification of expressed antigen by affinity chromatography. And then we have quantified the purified protein. And this purified protein has been used. So coating for the microbead with purified proteins to be used for the testing of TB patient or healthy samples. Here we have the results. We have done time course study based on how much time, what with the maximum time where we get the maximum production of this particular expression of this particular gene. This is only we are showing as a what we say representative results. And then we have, so in this case we have figured out from here we can use an induced, uninduced. So we have used this molecule, that is IPTG, for the induction, and as a result of that we get the expression of this particular antigen. And then so this is the time course study. Then we have done same after getting the time maximum where we can get the maximum expression. Then we have also used different concentration, so that is the IPTG concentration studies using the same protein. And then so in that way we have been able to know that which concentration we can use for the for our further experiment. So then we have gone for the bulk production of bulk expression of the protein, and then after that we have gone for the purification. So affinity purification we have done, and we have taken in each lane we have a specific sample. For example, here is we have sonicated sample, a filtered supernatant, and then flow through in this case, and then in the first case then again binding, 1x binding buffer wash, and here again 1x binding buffer wash with using some imidazole, and then we have wash again the buffer with imidazole different concentration, and then lane 6 is showing 1x wash buffer with 150 imidazole. Then lane 7 is showing, these are the lane where we have got these is proteins, in a way you can say purified protein. So we have eluded that by using the imidazole. Here is the marker, and in 9th and 10 has been used as a what we call this one has been used as a control, and here we have the sonicated pellet which we have got. And the molecular weight of this protein was 32.5 kd. Then we have confirmed that this is the right protein which we are looking for. So we have gone for the Western blotting. So here the Western blotting 1, 2, 3 is showing the different fractions of the same protein, and then fifth is the one here we have, and fifth is the one where we have the cell without plasmid, and then here we have the marker. So this is the one which we were looking for. So we have confirmed through Western blotting that we are getting at least the expression of this particular antigen. Then we have done the profiling, antibody profiling of reactivated TB patients, individual in multiplex microbead immunoassay, MMI. We have about 5 patients in this particular, and here the result is of 5 patients. And we have different antigens, AG85A, B, and C, and then we have CFP antigen 10, ESET antigen 6, HSPX antigen. And then what we have found, this plus, what does this means? So that 1 plus means low positive, 2 plus means medium positive, 3 plus means high positive. So we have seen, we have found at least in the 4 patients, this one positive, A, B, and C positive, as well as HSPX positive. And in the 5th one, we have got these 2 positive and then HSPX positive. What conclusion we have drawn? That is, the promising data showed that various expressed and purified antigens, when tested in multiplex assay, have potential to detect antibodies against these antigens in TB patients. So this study provides a base for the development of multiple antibody-based diagnostic test for detection of MB infection in active, reactivated, and latent TB patients. And then our studies are already in process. Then we have the next part of my talk, that is the DNA vaccine. Currently, it is unfortunate that having so many high throughput technology and so much information, we have not still being able to produce a specific vaccine which can be used for especially the adult pulmonary TB patients. So currently there is only one more than 70 years, about 70 years old licensed vaccine against tuberculosis, that is Bacillus calmette-guerin, BCG. This is live attenuated animal cell line passages. So this vaccine is available, and this vaccine is very good in protection of the children against TB for up to 10 years, but its efficacy declines with each subsequent year. So thus there is a need to develop a new and better TB vaccine to protect against adult and pulmonary TB. Generally, Modern vaccines typically consist of either killed, inactivated, in other words, inactivated. So how do we get this? By killing the disease-causing microbes with chemical, heat, or radiation. Then we have live, that is attenuated, and it is the version of a living microbe that has been weakened in the laboratory so it cannot cause the disease. Then we have subunit vaccine. Scientists thought that rather than using the whole organism, we can use the component of the organism, which are the antigenic. So that's why they have gone to the subunit vaccine that use components of pathogenic organism rather than the whole organism. Then they realized that rather than going for the whole component or whole protein, we can use the peptide vaccine. We can make the peptide vaccine based on the short peptides that mimic epitopes, that is antigenic determinant. So rather than having the whole protein, we can go only for the small peptides. So that is the peptide vaccine. So DNA vaccine, and then they have realized that it's better to go for the DNA vaccine because in some cases, sometime it happens that peptide vaccine and subunit vaccine, they are not properly working in all the different cases. So we have another opportunity to be used, another option we have to be used or to be established. DNA vaccine, the gene encoding an antigenic protein is incorporated. So we take the gene. Gene is the one that is injected, and then the antigen will be synthesized, and so in that way we will be able to avoid many other problems which we can see in the peptide vaccine or in the subunit vaccine. Thus, we also focused on the to develop novel, well-defined vaccines based on genetic information of the MTB. And why? Another reason was for that because DNA vaccine stimulates cellular and humoral immunity. So for this, we will go to these figures. This figure is showing the basic concept of the DNA vaccine. You have a gene, and then gene, of course, it is a DNA in this case, and then that will produce the RNA, and then RNA will give the protein. That protein is actually, we have taken the gene which has the antigenic properties. So what we do then, so gene for an immunogen, and then for an immunogen, insert gene into an expression plasmid, transform bacterial cell, and then grow bacteria. Then we will purify the plasmid DNA, and then this plasmid DNA will be used to immunize the rabbit. So immunize with immunogen expressing plasmid. So we immunize the rat, and then we see the result, what happens. And here is the mechanism of plasmid uptake and antigen processing. Plasmid will taken up, that is a transfection, monocyte, and antigen presenting cells. So on one side they will activate the T cells, and on one side it is T cell selection and stimulation, and as a result of that we are going to get the cytotoxic T cell response through the T helper cell, helper 1 and helper 2, and the other one is the B cells stimulation and selection. In this case we are going to get the antibody dependent immune response. So in other words, it is going to neutralize the Mycobacterium tuberculosis. So here we have experimental strategy, in this case selection of 6 immunogenic genes we have taken based on our also the data which we have got for the multiplexing and diagnostic procedure, and then we have used PCR hot start technique to get more of these, and then cloning into the specific vector as a simple vector, and then expression vectors, and then we have done the sequencing to make sure that during this process we have got the exact sequence which is present in the native protein, in the native gene, or in the native protein. Transfection and then expression, then we have used these for the transfection in expression in 293 T cells. So this is the in the cell line, and then second one we have immunized the BAP-C mice to see what results we can get. Animal trial studies have been done. So BAP-C female mice at the age of 8 weeks we have used. Different number of animals have used for different kinds of antigens. So these are they have been vaccinated by HXPS, CFP, similarly for ESET, AG85A, BC, and then we have also taken as a control positive control as well as the negative control. Then we have done the antibody testing. Animals were bled at certain times after post immunization. Then antibody was confirmed through two procedures: Western blotting as well as multiplex microbead immunoassay. So here we have the results. This figure is not that good. The transfection of MTB gene clone in 293 T cells. Cell line based expression were checked and antigen specific antisera were used to confirm the expression. So here we have the results of the HSPX that has the molecular weight about 50. So this is the purified one, then we have the transfected ones, and then we have the markers, and then we have the negative, what we call this, as a control, negative control, and then we have positive control, and here we have different concentration of the different concentration of the DNA for the transfection or for injection. So then we have used this immune response, immunoblotting for the same, that is HSPX PND vaccinated animal. And we have taken that sample, and then we have used this for the to see the immune response, and then it is the confirmation of the vaccine HSP vaccinated one, vaccinated animals, from where we are getting the positive result. It is as usual is our positive control, and here is the markers. Then we have, and the molecular weight was about 16 kD. Then we have also done the multiplex microbead immunoassay response to see in the vaccinated animals. And here we have used the same antigen. Antigen, that is the same gene, in which is going to be available in the form of antigen in the body, and then that is what we call the DNA vaccinated animal. So this is representing DNA vaccinated animal, and non-vaccinated animal we have used this color, which is not here. So this is the non-vaccinated, and here is the vaccinated. Each animal has given different what we call different responses, but they all are able to respond as compared to the normal. So with this, similarly, we have gone to have that is the vaccinated and non-vaccinated. This similarly vaccinated and non-vaccinated. This one vaccinated and non-vaccinated. And here these are the number of animals. MAB response from DNA vaccinated animal. The antibody response from these A, B, and C. Vaccinated group, lane 1 and 2 are vaccinated animals. Each animal is providing response against each vaccine inoculated in here. 3 and then 4, these are the negative control. Okay, so that is how we have been able to see that in the vaccinated animal they have been able to give the response. Based on these data, we have concluded that the initial results demonstrate that DNA vaccines are efficient in producing antibodies, and it could be a good alternate for conventional vaccine. It can be used as a single, like in the case of HSPX and CPF, which result we have not shown, or in the combination like a cocktail vaccine AG85A, B, and C. The elicitation of immune response against these well-characterized antigens can be very helpful in reducing load of MTB in patients alone or in combination with other therapies. So that is what we are expecting, and we believe that one day whatever groups are involved in this particular in this particular research to get a very conclusive objective, so they will be able to one day it is going to be more effective procedure available based on multiplexing in terms of diagnostics, and similarly based on multiplexing in terms of the vaccination. And here is the team of the scientists who worked for this procedure. It was actually HEC US collaborative grant which has been provided to us. So here is our group from the Pakistan, and here is the group who worked in the USA. So we had the collaborative project, and then thank you so much. And with this, I believe that we have a number of scientists, women scientists, who are very energetic, and they are also attending the meeting. So I have just one. It's not my what we say, my advice. It is saying by a very important person of the world, Nelson Mandela. He says that action without vision is pointless. Vision without action is fruitless. But with action and vision, you can change the world. That is what he did in his country. And similarly, our team, Pakistan chapter of OWSD, will be able to at least to encourage our new generation to take part in our STEM, and one day they will be able to contribute their share in this task, which our young scientists along with us are taking part. So thank you very much, and I stop here. And I'm sorry because it is recorded, so I cannot take any question at this point. But if you have any question, you can contact me. I will be very pleased to answer the question, and I'm really exciting for this particular program, which is one-day program, one-day conference, and hopefully it will be very successful, and we will get many ideas how to run in future. Thank you very much, and thank you for all our team. Thanks a lot. Bye.
That was an insightful discussion by Professor Azra Khanum.